Hello everyone, welcome back to my channel Xgene and ProteinX. In this tutorial video, I'm going to talk about the overview of the antigen presenting cells, and mainly I focus the signaling and the different kind of receptor of the antigen presenting cells. Let's begin. So antigen presenting cells are the special kind of immune cells that process and present antigens for recognition by T cells. So as you see in this figure that antigen presenting cells it present antigen by the Msc molecule or major histocompatibility complex, and that antigen it recognized by the T cell receptor or of T cell, and then T cell get activated along with by other stimulatory factors. So there are the classification of the antigen presenting cells. It classified into two groups, professional and non-professional. The professional antigen presenting cells express major histocompatibility complex 1 as well as 2. Other hand, the non-professional antigen presenting cells it express major histocompatibility complex 1 only and coupled to beta microglobulin. And another big difference in between these two groups, that is the professional antigen presenting cells are very much specialized to process and present antigens compared to the non-professional antigen presenting cells. So the professional antigen presenting cells included macrophages, dendritic cells and B cells, whereas the rest of the other nucleated cells all are non-professional antigen presenting cells. The location, the dendritic cells it locate skin, mucosal epithelium, lymphoid tissue and connective tissue. Macrophages it locates lymphoid tissue, connective tissue, body cavities, and B cells it locates blood and lymphoid tissue. So the antigen presenting cell first recognize and bind their target. To do this kind of job by the antigen presenting cells, it express antigen specific surface receptor including pattern recognition receptor or PRR. So one of the major family of the PRR is a toll-like receptor, that is TLR. Here it is showing, and TLR typically expressed on the cell surface or within endosome and type 1 transmembrane proteins. So it bind and recognize different kind of extracellular domain, that is one is pathogen associated molecular patterns that produced by the microbes, as well as it also recognize and bind with the damage associated molecular patterns. In short, it is known as DAMP, and it is produced by the damaged or mutated host cells. And once the extracellular bind domain its target, then the TLR it activates a cytosolic signaling cascade, which is initiated by an adaptor protein that interacts with the intracellular domain of the TLR. And depending on the TLR, two adaptor protein sets can be activated, and these are the TI-RAP mid 88 and TRAM-TRIP. So these are the two adapter sets that are required to activate the signaling cascade by the TLR. So another kind of PRR or another kind of receptor of the PRR member is the nucleotide binding oligomerization domain like receptor, that is known as the NLR, and NLR present in the cytoplasm, and like TLR, NLR initiate signaling cascade upon binding with the pathogen associated molecular patterns or PAMs. So next is the Msc molecule. Msc1 receptor are produced by all nucleated cells and display endogenous antigens. So MSC11, this molecule only present endogenous antigens, and to activate the CD8 cytotoxic T cells. So whereas the APC, it expresses both MSC1 as well as MSC2, and this MSC2 it only present the exogenous antigen, and by these exogenous antigens of the MSC2 it recognize the CD4 T cells, and then CD4 T cells get activated with other stimulatory factors. So only the APC it can activate the CD4 T cells, whereas all kind of other nucleated cells they can activate the cytotoxic T cells. So I'd like to mention that the macrophage, that one kind of antigen presenting cells, that when recognize the microbiota or food antigens, then it maintains tolerance. So upon binding appropriate PAMP or DMP, antigen presenting cells internalize their target by initializing phagocytosis, pinocytosis, or clarithrine mediated endocytosis. So let's have a look at how antigen presenting cells it process and present the pathogens. So the antigen presenting cells, when recognize any bacteria by the TLR receptor, and then it internalize by phagocytosis process. So it produces vacuoles, and then by the help of the vacuoles, bacteria get inside, and that vacuoles also it associated with some lysosome that forms the phagolysosome, and that bacteria antigens it digests by the lysosome, and it disintegrated into smaller pieces. So usually the major histocomponent complex or MSc molecule it present near the endoplasmic reticulum, but upon digestion of the antigens of bacteria by the lysosome, then this major histocomparative complex it associate with that antigens, the smaller pieces of the antigens, and then it ready to display on the cell membrane. So whenever it gets or it associated with that antigen, and then it display on the cell membrane or cell surface, and then this antigen it recognized by the respective T cell receptor, and then T cell get activated with the help of other co-stimulatory factors. So this is all about the antigen presenting cells overview. So I hope this video will be helpful. If you like this video, kindly hit the like button, share it, and please subscribe my channel. Thanks.